Well, hi. How you doing? For some of you here this afternoon, you may be asking, why does science matter? For others, you may be asking, what has science done for me lately? I respond with lots. Hello, my name is Asher Hamilton, and I'm the coordinator of student engagement here at 4Cs. Today, we're going to be wrapping up Latinx Heritage Month with the renowned researcher, scientist from Yale University, and you'll hear more about that. Today's program is a collaborative effort with the Office of Student Engagement and the 4Cs STEM Starter Program, and you'll hear more about that as well. So, if you believe that science or scientific discovery doesn't amount to much, I would say, think again. Science impacts every single one of us every day of the year. From the moment that we take our first breath until we take that last breath at death, science matters. From the electronic parts that interact with the computer boards on your car to the Zoom technology that's being read and fed into your computer, science matters. Think about how your cell phone integrates how pictures, calls, and text are fed from a satellite and back into your system. Think about that prescription of antibiotics that treat that sore throat. Think about the clean water that comes from your faucet and the lights that you ask your Google Home to turn on and off. This is what science has created. Now, as our world continues to change and become extremely, exceedingly complex along several dimensions, whether it be socially, politically, ecology, medically, or digitally, it's important for science teachers to provide students with the skills, the knowledge, and the ability to apply their learning to a real world around them. It's important that teachers empower their students with the agency and the tools to meet the unpredicted and unforeseen of tomorrow. Now, think about when JVC, back in the 1970s, that is, created a VHS. Do you think that they anticipated that consumer-oriented storage would take place in a global storage cloud? You see, often teachers assume that their students aren't capable of accomplishing big things. It's true that there are some students who've come from communities that have been excluded. Think about immigrant communities, ethnic minority communities, and students that may come from low income resource schools and neighborhoods. Think women. For science to be truly used as a tool to improve the human condition, the entire human condition, that is, we must develop an awareness of the current and historical injustices and injuries that have been promoted or perpetuated by science and scientists. This is truly where you can see a marriage between science and social justice. So, like I said before, we're going to hear a little bit more about who our renowned scientist is from Yale. But let's hear a few words from Bridget Berger with regard to the STEM Starter program right here at 4Cs. Bridget?
Thank you, Asher. Good afternoon, everybody. My name is Bridget Berger, and I'm the Director of STEM programs at Cape Cod Community College. I'm honored to welcome Dr. Colón-Ramos here on behalf of the STEM department and STEM Starter Academy at Cape Cod Community College. In partnership with the Massachusetts Department of Higher Education, my colleagues and I at 4Cs are dedicated to supporting students in STEM pathways. Whether your goal is to earn a STEM degree or certificate, transfer to a four-year school, or launch a career in STEM. We have a variety of programs, and I encourage you to email me and I can walk you through the different ways to get involved. Through STEM Starter Academy, the NSF-funded STEM Scholars Program, and the Cape Cod Regional STEM Network, we are working collaboratively with partners, both on and off campus, to create a STEM learning ecosystem in our region that goes from kindergarten to career. It fosters student success. We expand access and equity in STEM for the traditionally underrepresented populations in STEM, and we seek to inspire the next generation of innovators. It is my great honor to introduce to you today a scientist, professor, scholar, and researcher who embodies success and innovation in STEM, and already in his career has earned remarkable distinction and recognition for his efforts. He is also an advocate and activist for equity and inclusion of minorities in STEM fields, in particular for Latinx STEM students and professionals. Dr. Daniel Alfonso Colón-Ramos is the McConnell-Duberg Professor of Neuroscience and Cell Biology at Yale University School of Medicine. Dr. Colón-Ramos was born and raised in Puerto Rico. He completed his BA at Harvard University, his PhD in the lab of Dr. Sally Kornbluth at Duke University, and was a postdoctoral fellow in the lab of Dr. Kang Shan at Stanford University. A fundamental question in neuroscience is how synapses are assembled in living animals to produce behaviors and store memories. The Colón-Ramos lab is focused on this question and uses the nematode C. elegans to examine the cell biological mechanisms by which synapses are precisely assembled during development, maintained during growth, and modified during learning to store memories. Professor Colón-Ramos has also been very active in helping his native Puerto Rico obtain both scientific and material support, particularly after Hurricane Maria in 2017 and during this year's COVID-19 pandemic. A nonprofit he co-founded in 2006 called Ciencia Puerto Rico, and its affiliated Yale Ciencia Academy also provides professional development and leadership training to PhD candidates from minoritized backgrounds, particularly from Puerto Rico. In 2020, he was named for his outstanding contributions to medicine to the National Academy of Medicine, one of the highest honors in medicine. So it is my great pleasure to welcome you today, Dr. Colón-Ramos, to speak with us. Thank you so much for being here.
Thank you. Thank you, Bridget, for that introduction, and Asher for the introduction of the program. And what I wanted to do today was to start by sharing my passion for science, just explaining a little bit what a scientist is and what we do and what I do in the lab. So I'm gonna share a short presentation that I put together. Let me open this here and walk you through it. Can you see my screen? Yes? Okay, great. So let me play this. So this is one of the questions that I'm fascinated by. How do animals remember? And I don't know if you can notice, but these question marks are actually worms. This is you can see it, the worm head here. And this is the body, that's an embryo up there. And this is just a mirror image of that same thing. So we actually use these animals, these nematodes, they're called. They're not that big. They're about the size of a comma in a sentence, to understand how memories form. And when I tell that to students that haven't worked in memories, they get confused. So I like to explain how and why we can do that. But I'm gonna start with an anecdote, which is that, I mean, I was, as was mentioned, I was born and raised in Puerto Rico. And when I go back home, I mean, I didn't come from a family of scientists, and my aunts and uncles kind of understand what I do because I've been explaining it to them for 20 years now, but not fully. So I get a lot of questions of what is it that I'm doing studying worms and things like that. And I also get asked a lot now these days when I get interviews with the press, I get asked a lot, when was it that I realized that I wanted to be a scientist? And the way that I choose to answer that question is to say that I think that we are all born scientists. And I actually did the experiment to show it. So you shouldn't repeat this at home. This is an experiment done by a professional, but I'll show you the data. So I'm, besides being a scientist and everything that was mentioned, I'm also the father of triplets. These are my daughters when they were a year and a half old. Again, this is a highly dangerous experiment to repeat at home. I don't recommend it. Having triplets to show that we're all born scientists, I'm not sure about in retrospect. But this is the very first time that they were at the beach. This is actually back in Puerto Rico. They're seeing sand for the first time. And notice how over here you have Keisani, which is like grabbing the sand and looking at it. And this is almost like a time course here, because you have grabbing the sand, looking at the sand, and here Beatriz is actually eating the sand. So they're looking at the sand for the first time, and actually scientists, not many scientists at least, put things on their mouth all the time, but we use our senses to learn and to learn about new environments. And I want to use this experience that my kids are having here in learning, in being curious about their environment, in exploring it, to explain how scientists learn. So the first thing that I like to emphasize is that how we learn is context dependent. So they are learning about sand because they're at a beach. If they had been in Connecticut on top of a big pile of snow, they would be learning about snow. So our context is important. I'll come back to that because context also has to do with identity. So here's another experiment done in a different day. This is in the fall. They were a little older. And we have Beatriz and Alexandra here, and Keisani's in the middle. They have pretty short hands when they're babies. So Keisani cannot reach out to the leaves that are nearby, but her sisters, Alexandra and Beatriz, could. You can see that they have their hands like full of leaves, and in this case, the mouth also. So they're now learning about leaves, right? But Keisani observed how her sisters were grabbing the leaves and eating them and spitting them out. So with that, I'd like to emphasize that scientists also, we learn from each other. So we don't have to actually go through the experience. Like in this case, Keisani didn't have to go through the experience of eating leaves to learn that they were not edible. She could see her sisters going through that. Similarly, the reason that you're in a place like the Cape Cod Community College is that you can learn from things that others have discovered in science. So we learn from each other. This is an important trait both for scientists and for scholars and students everywhere. So we don't have to actually rediscover the wheel every time. That's how we make progress through science. And also very importantly, this is like many years later, they're much older. Actually, my daughters are 10 now. So they're not eating leaves and sand as much anymore. So this is many years later, they're in a pile of leaves and note how they're not eating them anymore. So what we learn is widely applicable. What I want to emphasize with that is essentially, they don't have to go to every beach in the world and taste every like grain of sand to realize that sand is not edible. You have one experience, you kind of draw from that experience, and then you apply it widely. And similar with the leaves. And I mean, I'm making a joke out of this, but these are important concepts of how humans learn that scientists also use. Like if you take something much more sophisticated like language, you can hear the accent in my when I speak English. If I was speaking in Spanish, you will hear a Puerto Rican accent, but you wouldn't hear the same accent that you're hearing right now. And the reason that I don't have an accent in Puerto Rican Spanish is because I was born and raised in Puerto Rico. But when I was a baby, although I don't remember any of it, when I was a baby, all babies that are, all of you, when you were babies, we used methods not unlike the scientific method to decode language, which is probably one of the most complex things that a brain can do. And everyone does it successfully. If you're, if you're a normal human being, you do it successfully as a baby, not knowing any language. So, and the way you do it is by using techniques not unlike the techniques that scientists use to be able to decode other complex aspects of nature. But here are three that I want to emphasize. We learn based on context, we learn from each other, and what we learn, we can then apply widely. So what I learn in worms, for example, I can apply widely. Why? Because the building blocks of an animal like a worm are not unlike the building blocks that we find in our own brains and nervous system. So I'll come back to that. But I want to emphasize now the aspect of like, we learn based on context. This was my context. So I grew up in the Caribbean. These are baby leatherback turtles that are hatching from a nest not far away from where I was born and raised. This is actually very close to San Juan, which is the capital. And note that all these turtles, they're coming out and they don't know what to do. They're all going in the same direction. You don't see any turtle here going to the right-hand side. And the reason is because the ocean is to the left. So they're all beelining it towards the ocean. And as a child, I will look at this and ask myself, okay, these animals haven't been to school. So how do they know where to go? Who taught them? Who taught the turtle to go towards the ocean? And the reality of it is that this behavior that we're observing here is an innate behavior. You can think of it like a memory that they have inherited from their ancestors. In reality, it's a behavior that is selected for by evolution, which we humans call instinctual behaviors or innate behaviors. Humans also have it. So there are a lot of behaviors that babies have, like for example, the ability to learn language. That is an innate behavior. Now, what language you learn depends on your context. But the ability to learn language is an innate human behavior. So you have innate, or we call them hardwired, because the turtle, like the turtle, it knows what to do. Like a baby that's also lactating, for example, knowing how to lactate from a breast. That's an innate actually an innate mammalian behavior. But I also want to emphasize that these animals are forming a memory. During that movie, those animals formed a memory, and they formed a memory of that beach. And those turtles, they're gonna grow up to be very large. I'll come back to that. And they will remember that beach. But right now, I want to emphasize that the innate behaviors, they determine during the development how the architecture of the circuits form. So essentially, the brain of this animal formed in a certain way that when it comes out of the nest, it knows where to go, but it also formed in a way that allows the animal to learn. So I'm interested in understanding how is it that this architecture of a brain, be it a turtle brain or a human brain or a worm brain, is both stereotyped, meaning it's built in a specific way as to encode for certain behaviors that are innate, but also is flexible as to accommodate for things like memories. So that's what I like to study. That's the question that fascinates me. Another way of asking that, when I was a child, I would ask it this way: how do the turtles know what to do? So again, I want to emphasize these animals. So they were born in Puerto Rico. Puerto Rico is in the Caribbean, in the middle of that circle. Cape Cod will be, of course, like around here. And they will travel the world's oceans. Actually, some of them, you might find them near Cape Cod as adults. They grow up to be about 9 feet long, about the size of a small car, like a Volkswagen. And they will travel the world's oceans. But when they have to make the most important decision in their lives, and for a turtle, a sea turtle, the most important decision in its life is where it's gonna lay its eggs. So when they have to make the most important decision in their lives, guess what? They remember that beach, they remember that memory that they formed during the first few minutes of their lives, and they'll come back to it. So that is a plastic memory. So the ability to know to go towards the ocean when they're born is an innate behavior, but the beach that they remember is plastic, because if they had been born in the Dominican Republic or in the Gulf of Mexico, they'll remember a different beach. So that is a plastic memory or a malleable memory that's dependent on context. Used by analogy language, our ability to know language is innate. What language we learn depends on our context. So, but how they learn that, how a memory is formed, how is it retrieved? How is it used to find the beach again? No one knows the answer to that. And with that, I'd like to emphasize to the students that some of the most fascinating questions in science lack answers. So I like to emphasize that, because the way that we teach science is that every question ends with an answer. And the way science is done is that every answer ends with a question. So if you have a question about something, it's not because you're dumb, it's because you're curious. Don't stop asking questions. Don't let anybody take away your right to ask a question. We know some things. It's not like we don't know anything. We know some things. We know, for example, that memories reside in the brain. Like that, we know that the nervous system is important. We know specific functions of the nervous system. We know those things, but no one right now can truly explain to you, when you think about the image of your mother, or your first kiss, or whatever experience it is that you had that makes you who you are, what that is. No one can explain that. No one on planet Earth alive right now, or no one that had ever lived in the history of humanity, can get on a blackboard and draw and explain to you what is a memory. Maybe you will be the one that figures it out. So one healthy human brain, just to give you an idea of why this is a complex question and why we don't understand it, one healthy human brain has 100 billion neurons. That's a whole lot of cells. And those cells are specifically connected into circuits that enable us to form memories. Just to give you an idea of how many cells we're talking about, there are more cells in your brain than there are stars in the Milky Way. So in those like really dark nights in Cape Cod, when you're looking up at the stars before it gets too cold, and you ponder about the eternity of the universe and the stars and all that thing, think that it's just but a reflection of your potential and all the connections that you can make and all everything you have in your brain, because it's no more complex than your brain is. So what do scientists do? Like, what is my job? Like, it's a question that I get a lot. What is it that I do? And I teach, like I give lectures like this, but this is not what I spend most of my time doing. What I spend most of my time doing is research. So what? And scientists, the way I will explain it is that they are producers of knowledge. So what does that mean? So I will say that students are consumers of knowledge. Consumers of knowledge because you go to college and you consume knowledge. Somebody stands in front of you and they share knowledge with you and you consume it, right? It will be like, it's the difference between like an artist that makes a song and somebody listening to the song. So you're listening to the song and making the song. So scientists are producers of knowledge. We also disseminate it the same way that the artist will write the song and then play it. So we disseminate it, and that's part of what I do too. I disseminate knowledge. And the way that we do that is by using these precepts that I introduced with the images of my kids. So we learn from each other, and scientists have conferences. I go to Cape Cod every summer, and it's not to vacation, it's to go to a center that is in Falmouth called the Marine Biological Laboratories, where I spend many weeks, sometimes up to a month and a half, meeting with colleagues from around the world that go to that world-class center that's located there in Cape Cod to exchange ideas, to learn from each other. And then how we learn is context dependent. So of course, my interest as a scientist, as a Puerto Rican scientist, of course, like what I'm learning is its fundamental aspects that are applicable to neuroscience anywhere in planet Earth. But my interest in neuroscience, I gave you an example, is rooted on my identity as a Puerto Rican, which dependent on my context because it is who I am. So it was experiences like the one that I shared with you of the turtles that led me to ask the questions that I'm asking right now. Not using turtles to study them, because we're talking about animals that cannot even fit through the door of my lab, but using smaller animals that are much more practical. But the knowledge, although my interests are context dependent, they're very me focused. What I learn about how the nervous system works is widely applicable. So, and that is true for all scientists. Like all scientists have a story of why they're interested in the questions that they're interested on. But the discoveries that they make are not specific to them. They benefit all of humanity. That's important to consider. So with that, I like to emphasize, and this is why I give this type of talks, where scientists come from matters. This is the importance of diversity in science, because where scientists come from influences what they're interested on. And what influences what scientists are interested on will influence what science gets done and what science doesn't get done and who gets to benefit from the science. So where scientists come from matters. Although their knowledge is universal, not so the impact of their science. So I'm interested in how you go from molecules to mind. How is it that molecules can build up what we call the mind? And here are three kind of grand challenges. This is just an animation of neurons communicating with each other, which is essentially what we look. This is a cartoon. This is not based on real images from a microscope or data. This is an artistic rendering. But here you have a connection between two cells that's talking to each other. And there are three questions that I'm interested in. How are brains built? So you have 100 billion neurons that are connected to each other very specifically. How? Like, people like to make analogies between brains and computers, but computers are way dumb compared to a brain. And that's one. And two, computers are built by people. So I can get an engineer and they can explain to you exactly where each circuit is and how they're connected to what and why is it that when you click on your keyboard, the letter A, your monitor displays the letter A. They can explain all of that. But guess what? We didn't build a brain. We don't have the faintest idea how a brain works. So I cannot explain to you how is it that right now I'm, controlling the vocal cords and communicating somehow, emitting sound waves that are moving hair cells in your ears and hopefully transforming into ideas and maybe memories. So no one can explain that. So how is it that you build a machine that's capable of doing that? What is a memory? And in terms of molecules and cells, what is it? How do we store it? Where is it stored? How do we recall it? And what happens when they break down? If you want to cure diseases like Alzheimer's and Parkinson's and neurodegenerative diseases in which you have memory loss, guess what? You better know what you're losing. You better understand what is a memory and what you're losing, because if you don't even know what a memory is, how are you going to find what you're losing in the disease? How can you fix a car if you don't know how the car works? The reason that we can fix cars is because we understand, the mechanic understands how the car works. But if we don't understand the engine, if we don't understand the brakes, if we don't understand the gas, if we don't understand the steering wheel, who's going to fix that car? So we need to first understand how it works before we can fix what's broken. That's something that I emphasize a lot as a scientist. That's what we do, basic research. That's what we examine things. And only if we understand it can we apply then the knowledge into something that's practical. That brings me to the system that we use to understand these questions. And that system is a worm that you're seeing here. This is the head. These are two eggs, so it lays eggs. This is the tail here. And when I give a lot of these talks and I'm talking about memories and remembering and vocal cords and all this beautiful stuff. And then I tell them that I work on worms and people are like, what? So how can you use a worm to understand a human brain? And this gets to the maybe most important thing that I'm going to say today, which is that when nature finds a solution, it uses it over and over and over again. If you want to understand how a tree makes energy or how a plant can make like a starchy vegetable or something like that, you need to understand how chlorophyll works. You need to understand how it traps like sunlight. And if you're going to understand that, if you understand that for the simplest algae in the ocean, you understand that for the giant sequoias. You don't need to go understanding chlorophyll in every plant to understand how it works. The same way that my kids didn't have to eat sand in every ocean to know that sand is not edible. Similarly, if I understand how this animal remembers, it's not going to form the same memories I form. But if I understand how it remembers, it's going to give me fundamental ideas of how my brain is capable of forming memories. And I can understand it far easier in an organism like this. Another example that I like to give is, guess in which system we have made most discoveries regarding cancer? Yeast. That yeast that you use to make bread and the yeast that you use to make beer and wine. Yeast. Yeast is a fungus, it's a single cell, and most of our understanding of cancer comes from yeast. Why? Because yeast are single cells that are eukaryotic cells. They're essentially cousins of our cells. We're multicellular organisms, but the mechanisms that a yeast cell uses to decide if it's going to divide or not are exactly the same mechanisms that our cells use. And guess what breaks when you have cancer? The ability of a cell to know if it should divide or not. When the cell shouldn't divide, it starts dividing. It starts dividing, it forms a tumor. It forms a tumor, you get cancer. The mechanisms that tell the cell don't divide are the same in a unicellular yeast as they are in a multicellular human. So if you understand those mechanisms for the yeast cell, you understand it for the human. Similarly, if you understand this brain, look, there are going to be things that I'm not going to be able to explain. These animals don't have eyes. So if I was interested in how eyes work, I'm not going to be able to study it in that animal. If I wanted to understand how I talk, I'm not going to be able to study it in that animal. But if I want to understand how two cells communicate and how that communication changes to store a memory, I will be able to understand that in this animal, and that's what we do. So this is essentially some of the questions that I get. Hopefully I've answered some of them. So people ask me like, how can you use a worm to understand a human brain? Because the answer to that is because when nature finds a solution to a problem, it uses it over and over again. So we can use evolution to be able to understand fundamental aspects of how cells communicate in this animal. Like why play with worms and not work on a disease? Because if you don't understand how things work at a fundamental level, you're not going to be able to fix it. We didn't make the human body. No one, if you don't understand how to do that, medicine without basic research is little more than witchcraft. Before basic research, doctors were bleeding people and measuring how big heads were to try to understand how people thought. That without basic research, medicine that cannot do much. And knowledge is cool and it is very useful because if you don't have the knowledge, you're not going to be able to apply it. So we do what's called curiosity-driven research. It's like research based on our own curiosity, but by doing that research, we come up with fundamental lessons of how things work that then we can apply widely. Again, nature reuses solutions to problems, so what works here works in other systems, meaning in other organisms, and what we learn is widely applicable. And one of the cool things that we can do in this animal is look at its neurons in the living animal. So here I'm showing you literally all the neurons labeled. You can see it almost looks like a machine. It's stunning. Here's the brain of the animal. You can see all these cables connecting the muscles and everything. And this is actually an image from one of my colleagues. The animal is transparent and we can light it up like a Christmas tree. So here we're labeling two different classes of neurons. For those of you that have taken some neuroscience classes, GABAergic neurons and cholinergic neurons. So how can we do this? How can we take this animal and just label it like a Christmas tree? Does it normally look like that? And the answer is no. Normally in nature, this animal is not fluorescent like this. So how can we make it fluorescent? And I wanted to explain this because this is a story that ties to the place that you're at, Cape Cod. So the way that we can do it is by applying knowledge that scientists have generated from other systems. So it turns out that many years ago, a Japanese scientist who later moved to the marine biological laboratories, I'll put his name here, Osamu Shimomura, he just became really curious about how jellyfish were bioluminescent, like or fluorescent. How is it that they emit light? And he decided to study that, and he discovered that essentially these animals are emitting light because they have two proteins that are capable of doing so. One of them is a protein called aquarin, which binds to an element called calcium, and that binding then leads it to emit blue light. And then there's a second protein that takes this blue light and converts it into green light. So these animals actually end up emitting green light. And that second protein that converts blue light to green light is called green fluorescent protein. So the people that discovered this, including Osamu Shimomura, who later worked at the marine biological laboratories, ended up winning the Nobel Prize. But there's another character, another scientist that was instrumental in this. And actually many of the discoveries were made not far away from where you are today, near Falmouth. These other experiments were done at the Hui by a person called Doc Prashad. And he was the one that actually purified the proteins that emit this light, and that was and then figure out the DNA sequence that was important for codifying those proteins. And that was very important because if you know that, then you can take those sequences, those instructions that tell the jellyfish how to make a fluorescent protein and put it into any animal, and now that animal makes a fluorescent protein. So you can use that same gene, take it from a jellyfish, put it into, for example, in this case, a mouse. And here you're putting fluorescent proteins of different colors, and they're lighting up this part of the brain, which is called the hippocampus, because it looks like a little horse. And you can see these beautiful ordered structures. And the reason we do that is not for aesthetics, it's because we want to understand how the brain is structured. What is the architecture of the brain? How does that architecture encode memories? And now we can actually see them. But note that the discovery, which again was recognized with a Nobel Prize, started with curiosity-driven research of a person just asking, how is it that this jellyfish emits light? So fluorescent proteins can label individual cells. So they're different colors, so GFP, YFP, M-Cherry, and you can express them in bacteria. So these are bacterial cultures that are expressing different fluorescent proteins, and you can see somebody very inspired drew a scene there of a beach. I kind of love this slide here. But also, more importantly, you can put them in live animals like we do, and you can label single cells. So this is the head of the animal. It will be right here. And that's the cell body. That is the axon. That is the dendrite. And that's how we can label animals like a Christmas tree. And then in our case, what we can do is we can, I'm going to just spend this slide teaching how this works. These instructions, these DNA instructions it's essentially a little, it's like a recipe that has 3 components. One of them is a genetic sequence called the promoter. And you can look this up, like if you can take a screenshot if you want, and then look it up online so you can learn how this works. You want to learn how the transgenes work. But essentially, this promoter is a sequence that tells the proteins or the genes where they should be expressed, in what tissues they should be expressed. So it has the instructions of where this protein that's encoded by the cDNA should be expressed. This has the instructions of the protein, and this has the instructions from the jellyfish with the fluorophore. So you can express any protein in that animal that now has essentially a light bulb. It's almost like putting a flashlight or a spotlight on the protein, and you can see it anywhere in the animal. So we have done that, and these triangles represent cells that are connected. Those arrows represent connections or synapses, and we can label those individual cells, which have these 3-letter codes. And this, to the right-hand side of my screen, you have the pictures of those cells in a living animal. So we can see how the cell looks, we can see proteins inside the cell, we can label them in different colors. And the importance of that is, and I'm gonna walk you essentially here through the process of discovery and just explain one of our discoveries. And I'm like 1 or 2 slides away from finishing talking so we can have time for questions. But essentially, here we're labeling a single cell by marking all of the cytoplasm so we can see the morphology of the whole cell. Just pay attention to this part. I'll explain this bottom part in a second. So then the same way that we're expressing that fluorophore in the single cell, we can express synaptic proteins. So these are proteins that localize to synapses. You can see that we're not labeling this part or this part. We're labeling this part, which I put a box around it. And there are little dots in this part, which you can see here. Those are synapses. Those represent the connections with other cells that you're not seeing because we haven't labeled them. This is how they look when they're healthy and normal. So then we can make mutants. And what we're doing is like, when we make mutants, it's like, it's like when a kid gets a remote control car and they break it to see how it works. And if you break something important, the car stops working, which is why parents get pissed off. But imagine now the kid had a ton of remote cars that they could break at random. They will eventually figure out how the car works. So that's what we're doing here. We break it by creating mutants, and then we get mutants like this. So note that in this animal, which is a mutant, you don't have that much signal over here as you did here. So the synapses are kind of gone here. So once we have that mutation, we can ask questions like, okay, what gene did we break that's so important? And where is that gene acting? And what is it doing? And doing those type of analysis that start by kind of like labeling the things, understanding how they work when they work, and then breaking them, we can put together stories like this. So here are 2 cells that are forming a synapse. So a synapse has 2 parts, a cell that's talking and a cell that's listening. And this synaptic connection is important for them to function. And when we started our study, we thought that the way that this cell knew where to form these connections, which are represented here with these bubbles, like the way that it knew how to do it is because it was talking to this cell, and this cell was telling it, hey, come over here, let's form a connection. It turns out that that's not the case. There's a 3rd cell that's a matchmaker, which is called a glia cell. And that glia cell is talking to both of them. It's like, imagine that this is like the person that gets these 2 to date. And that glia cell is releasing a signal. In this case, it's called netrin, but if you don't understand what netrin is, it doesn't matter. It's a molecule that's very important for neurodevelopment. So netrin, released by the glia cell, tells this cell then it activates what is called a signal translation cascade, which is a bunch of proteins that end up reorganizing the cytoskeleton and forming the synapse. And that's how those synapses they're formed. So if you break any of these parts of the pathway, any of these proteins, you disrupt them, then you end up with a defective synapse. And then the concepts that we learned was that glia are actually the masters of these synapses. They're the ones that are governing, they're telling these cells where to form these synapses, both in early development, but also as animals grow, because animals grow many times, they're born as miniature versions of themselves, and then they have to grow and they have to scale proportionally. And no one understands, hey, how do you scale proportionally as you're growing and things don't break inside your brain. And we learned that what's holding it together are these glia cells that are kind of keeping these sites where the synapses are supposed to be as the animals are growing. So what else have we discovered? We discovered how neurons find each other and connect, how they maintain connections during growth, how they modify them during learning. We have a number of studies about that. And then how they keep them healthy and working well, and what happens when they break down. Actually, today I was just working on a study that I think is going to be very important for understanding Parkinson's, which has to do with a mechanism that we identified, which we think breaks in patients that suffer of Parkinson's disease. And we identify them in worms, but when we look at what's known in humans, it makes sense that this mechanism is actually breaking in humans. So many of the mechanisms are essentially is a term used in science to refer to processes. Many of the processes that we discovered in the past 10 years are also important for human biology. With that, I want to finish by showing you a picture of my lab. So my lab is pretty diverse. It has people from 5 different continents, 13 different countries, all over the US, all sorts of socioeconomic, racial, etc. And this is just one iteration. I've had, many people have rotated through my lab. So, so this is just the part of the current lab. And I'll stop there, and I'll be happy to have a discussion about the other topic or about what I just talked about. Thank you.
Thank you so much, Dr. Colón-Ramos. That is fascinating. In this next section, we're going to have several questions that Asher and I will ask first. And then we'd like to open it up to questions from the students and faculty who are on this call. So if you would like to submit your question for Dr. Colón-Ramos, please send me a message in the chat, direct it to me, and then we'll ask those questions in just a moment. So I'll start with my 2 questions that I'm going to ask, which are about you and your experience so far. Can you share, Dr. Colón-Ramos, can you share a turning point or a defining moment in your work, or I would also add in your pathway on becoming a scientist? Can you share about a defining moment? Who was involved in that moment? What did you do right? And why was it a defining moment? Can you talk to us about those defining moments?
I think a defining moment for me was when I worked in my first research lab full-time. So I had worked on labs before part-time, and the work seemed kind of distant and hard and disconnected from who I was or what I was interested on. I wasn't really understanding what the lab was working on. And then I decided to just plunge in and go to a lab where I worked full-time. And I worked full-time after graduating from college for about a year, just to really immerse myself in the research and see if it was for me. I highly recommend students to do that if they can. And work in a lab if they're interested in science for a year or even better, 2 years. Sometimes students think that it's, like a gap year and I'm like, a gap from what? There are no gaps in life. You know what I mean? It's just part of your experience of training and growing up. But anyway, so the reason that it was defining for me was a couple of reasons. One of them is I picked a mentor that, at the time I picked a mentor that looked like me, was another Puerto Rican professor. I feel like I needed at that time somebody that kind of looked like me that could help me think through my role in science. Frankly, he was very different from me. I remember talking to him about my identity and what I was kind of wrestling with. And he was like, oh, I don't have that problem. So it's not like he wasn't identical to me just because we shared the same background. But I needed to see somebody that shared my background doing science and see how they did it. So that was important. The other thing that was important for me was just to immerse myself, because I feel like I was doing science at that time like I was doing other extracurricular activities. Something that kind of you clock in at a certain time, you're there when you're there, and then you clock out, and then you don't think about it. And science, it's more like reading a book kind of thing. You can read a book and, you can spend you can go to a cafe and start reading a book and read 10 pages. And then you were really listening to the conversation that was happening next to you. And you haven't registered those 10 pages. You have to go back and read them again. So it's easy to be in a lab and be in a lab like that. Like you're kind of like doing things and moving your arms and kind of learning a technique, and students think like, oh, just because I know the technique, I know what I'm doing. And it's not any more than just because you know how to drive, you know where you're going. You know, you need to know how to drive a car, but that doesn't mean that you know the directions to where you need to go. So science has that. It's very similar to driving. You need to know the technique, of course. Knowing the technique is not sufficient. You need to immerse yourself. You need to look around. You need to understand the landscape. You need to know where you're going. And I feel it's very hard to do that. Like if you're volunteering like 5 hours a week or 10 hours a week, or just kind of like washing dishes here and there. So that was, I think, maybe a relatable experience that was transformative for me and that I suggest to students.
Great, thank you. Next question. So is there controversy in your area of research? What has been the greatest pushback? And I guess I would pause there, and then if we have time to ask, what are other schools of thought? Do you have, you mentioned you come to MBL and discuss with your colleagues. Are there really varying schools of thought around your work? Can you talk about that? But specifically, is there controversy around the work that you're doing in this field?
There are, but it's not like in the movies. I feel like, so it's not like in the movies where you just kind of like, it's not a political debate. You know, it's not like you take a position and you're like, okay, I'm just like taking, it's not a rhetorical argument. We're genuinely, like scientists for the most part are genuinely interested in finding something that approximates the truth, like how it really works. So many controversies are not about who's right, but collaborating in finding what's right. So it's not so positional, you know what I mean? It's not like you're not there to convince the other person just because you want to like save face or you want to be right. Like science is, that's one of the things that I love about science. When I was a student, I thought science was about what you know, and it's not. Like science is about what you don't know. So what you know is the trailhead. It's like, you know, when you go to a park and you're hiking and you take a road and then the road ends and the trail starts. So think of what you know as the road and think of what you don't know as the trailhead. So the road is only useful to bring you to the start of the hiking. So if you're kind of on the road, kind of like rolling on the road, really happy that you're on the road, being like doing like snow angels, being like, oh my God, this is awesome. Like I'm an expert on this road. You're not discovering anything. You're just on something somebody else built. Like science is about the unknown. It's about discovery. It's about self-discovery, and it's about asking questions. It's about being humble. In science, in real life science, every answer ends up with a question. It's really hard to explain to people because they're used to the other way around. All knowledge is incomplete. All knowledge is incomplete. Like you think about an atom and you're like, I understand that an atom and you can describe it, orbitals, yada, yada. Guess what? They're subatomic particles. Then you understand that. Guess what? They're particles that make those particles. And all knowledge, all frameworks are incomplete. And that, to some people, that sounds really disconcerting. They're like, oh my God, what does that mean? And it's not relativism, like, oh my God, everything is relative. I'm not saying that. They're real truths. But those truths, you live with the knowledge that you're constantly pushing the boundaries of ignorance farther away, and there's always more to do, like further to go. The best scientists that I've met in my life, and I've had the privilege of, you know, occupying spaces that are occupied by like Nobel laureates and some of the most accomplished scientists, they're not flaunting what they know. They're always asking questions about what they don't know. When I find a person that's flaunting what they know, they're usually not a good scientist. They're just somebody usually insecure that's just trying to like position themselves relative to somebody else. That's what I found. So these debates and these controversies, I invite them. I actually go and seek them out. Literally what I was doing before I got on this Zoom call was writing an email to two colleagues, one in Germany and another one in Utah, explaining a discovery that we had and putting it to them so they criticize our discovery. I want to know what is it that they find problematic about our discovery, because that's what's going to push me to make the discovery better. Like, I think, you know, when I was starting as a student, I didn't want that because you feel vulnerable and kind of like you just want to be right. But in the space that I'm in right now, it's the opposite. I'm I'm not looking to be right. I'm looking to be wrong. Not because I want to be wrong, but because if I rather somebody point that out because that's what advances my work. When I've had writings and I've sent it to colleagues and they send it back to me and they're like, oh, this is beautiful. It makes perfect sense. I don't send them my writings anymore. I think that's a waste of time. Seriously, it cannot be perfect. There's so much we don't understand. So how can it be perfect? So if you're telling me it's perfect, you just didn't read it or you're not thinking about it. Like I want them to be critical. I want them to go in there and be like, no, this is this needs to be more polished. You haven't thought about this deeply enough. What about this other question? You're not seeing that. That's the feedback that I'm looking for. So I'm looking for controversy, not because I love being wrong any more than anyone else, but because I know that if I'm not getting that from my colleagues, then it's there. I'm not finding it. It doesn't mean that it's not there. It just means that I haven't put myself in a position to grow. Like what I tell students, maybe if you're gonna take a lesson out of what I'm saying, is if you're in a room where you're the person that knows the most, you're in the wrong room unless you're teaching.
I think it's so important what you're saying, and it has so many applications toward K through 12 education as well. The way science is taught, and the way it needs to be taught to really foster that curiosity and willingness to grow and learn and fail and experiment. I mean, that's really, I think, the embodiment of that scientific process, which we can really bring in more to K through 12 education.
And that's how we learn. We don't learn by succeeding, we learn by failing. Like I mean, we don't want to fail all the time. Like that's not what I'm saying. And we don't want to be complacent in failure. There's such a thing as complacency in ignorance and failure. That's not what I'm talking about. I'm being more nuanced than that. I'm saying the way that you learn is by failing. Like the same way that the same way that we can see, because we can see light, but also shadows. If we could only see light, we wouldn't be able to see anything. We would just see one bright spot and you wouldn't be able to tell anything. Mm-hmm. But the same way that we produce knowledge is through failures. Failures give us the contrast between how things work and how things don't work. If everything worked the same, we wouldn't be able to learn anything. So if everything you wrote in your life, every essay, everything, you gave it to a teacher, and every teacher that have ever gotten it said, this is perfect, believe me, your writing will suck.
I'm going to turn it over to Asher for next questions, and then we'll come back for this audience questions that I've got in the chat. Thank you so much.
Hey, thank you, Dr. Colón-Ramos. I have the harder, a pretty interesting, but I would consider a question around attraction, retention, and diversity. It has been well documented that students of color are underrepresented in STEM professions and in STEM degree and certificate programs. Can you explore this fact as it relates to white privilege in STEM and how that privilege impacts communities of color in STEM, especially around the K through 12 sphere? In what ways do you observe the ways of knowing and the ways of doing in white cultures that are recognized and reinforced in STEM education? How can we help our students of color because of this, who are disenfranchised and discouraged in educations around STEM?
Well, so there's a lot there. Let me take it by parts. I've had students, and I recently had a student that was asking me. He was saying, "Look, I'm Mexican, and I'm just wondering how much of me being Mexican can I bring into the lab?" And I was like, "What? Why do you feel that you're gonna bring all of you into the lab, right? Like, why is it that you feel that you cannot bring all of who you are into science?" And that is a problem if academia or science or our education system is messaging to the students that they cannot bring all of who they are to the lab. And I've never had a white student ask me the same question, right? So I started reflecting about that. And one time I was giving a talk to a big auditorium. There were probably like 200 students and faculty. And I asked a question. I said, you can hear my accent, right? And everyone laughed. And then I said, okay, so raise your hand if you don't have an accent. And I would say about maybe 2/3 of the audience raised their hand. And then I repeated the question. I said, raise your hand if you have an accent. And then about half of them, I kept on repeating it until everyone lowered their hand. And I said, you might have an American accent, but you have an accent. And somebody said, no, but I have an accent like in the news. And I said, which news? BBC, Australia, CNN. Everyone has an accent. But not all accents are valued the same. I've had colleagues from India being told that they need to reduce their accent. And they speak English as a first language, because in India, English is a language that people speak as a first language. And I've had colleagues from France where everyone praises their accent. It's so beautiful. Accents, everyone has an accent, and not all accents are valued the same. That's a way of exemplifying culture in science. Everyone has a culture. Now, the culture in science, because it has, because of systematic barriers that have been established that have nothing to do with science and everything to do with society. The culture in science is a white culture. That is the culture in science. So when I have a student asking me, can I bring my culture? Because they make an assumption that science doesn't have a culture. It's almost like you have a white, like it's like not having an accent, because you speak like the majority, therefore you don't have an accent. You have the culture of the majority, therefore you don't have a culture. That's not true. There is a culture. It's a white culture. So not accepting other cultures into science is a form of white supremacy, because it has a culture. It's a white. So we need to create these spaces for other people to also be everything they are in the context of science and contribute to science. That doesn't mean that they're gonna make up how science is made. Like, that's not, like, I mean, I hold every person in my lab to the highest standards and the strongest rigors, but I allow them to be everything they are in the lab, because I tell them, I want you to bring all of you into the lab. And I believe, and I think I have evidence, I haven't done a real experiment or quantify this, but I've seen it, you can see it in my success, that when you do that, then people do their best, because they feel comfortable being who they are, and they bring their ideas, they bring their knowledge, they bring their curiosity. If you want people to innovate, you have to create environments where people feel safe innovating, which means that they have to feel safe failing. And it's the same with learning. So when you talk about K through 12, if you want kids to be learning, they have to feel comfortable asking questions. And if they they are only going to feel comfortable asking questions if they feel secure in that the questions that they're asking are not going to be judged. They're not going to be judged as other, and stereotypes are not going to be thrown at them based on the questions that they're asking. So if you don't foster an environment like that, you're not going to foster a learning environment, be it in the top universities or be it in a classroom anywhere in the world. So that's what I believe.
Thank you so much for your answer. What I'm going to do is surrender my time to the audience, because I think this is about our audience this afternoon and what questions that they have.
So here's a question in the chat. If we can't explain what a memory is or locate it within the brain, why are we so sure it resides in the brain? I understand that there are a number of neurological diseases that rob a person of memory, but couldn't the brain be a correlate of memory and not the seat itself?
That's a great question. So there are these really famous cases, one of them from Connecticut, of lesions in the brain where people lose very specific abilities. So there's a part of the brain called Broca's brain, where if people lose it, they lose very specifically their ability for language. There are parts of the brain where if people lose them, Oliver Stone, I think, had a number of books written about this. Like, if people lose them, they lose the ability to form, like that movie Memento is based on some of these patients, where they lose the ability to form short-term memories. So they remember everything in the moment, but they cannot store it. But they can form memories before they had the lesion that they had. So our current model or hypothesis of how memories are formed is that they're formed through changes in these connections between cells that alter the information flow, and those connections are present in the brain. Now we have those connections, and we have neurons all over the body. I mean, they're controlling my lungs right now. They're controlling my autonomic the autonomic nervous system, my heartbeat. I'm not thinking, let me control my heartbeat. That's all being controlled. But the difference between those synaptic connections and the ones that I'm talking about that are in the brain is that those we cannot modify. You cannot, it doesn't matter how much you train, like the reflexes that we have, for example, the reflex of like when they hit you in the knee and your leg goes up, you cannot control that. When you start thinking about it that way, you get a little bit of vertigo because you start realizing how much machine like we are in some ways.
Thank you. Next question. This is asking, what barriers did you encounter when you started on your path in science? And if so, how did you overcome them? And do those barriers still exist for others who are coming up in STEM? How can those students overcome that?
Yeah, so first I'll start by saying I obviously have had like a rosy experience, right? I have survivor bias because I'm a professor. So mine is a story of like whatever barriers I faced by definition, because I'm giving this talk today, I overcame. I'm sure there are people out there that might have faced the same barriers and have had different outcomes for different reasons. So I'll start with that because I want to contextualize what I'm about to answer. I have had, circumstances in which I felt I was, judged by how I looked or how I talked or where I come from. For sure, many. But I choose not to talk about those as much because I feel like, in science as in life, we use frameworks and narratives to explain things. And if that was my only narrative, I would be doing a dishonor to all of the people that helped me get to where I am today. So I will say, for me, for every person that I had that doubted me, I had 5 to 10 people that helped me along the way. And I have found that when I get deep into with students about those conversations, like you'll remember, most of us will remember, and that's how our brains are wired, the negative experiences, right? Like if you go to a restaurant and you have 20 meals and then you have one bad meal and you have food poisoning, you're not gonna go back to that restaurant if you remember that. But you had 20 good meals there, but which one you remember? The food poisoning one. Now that's fine because that's just a restaurant. But if you had 20 people that spent a lot of time mentoring you, talking to you, helping you along the way, and all you're gonna talk about is the one person that didn't do that, you're doing a dishonor to your family, to the people that came before you. To I don't want to frame it only about that one narrative. And in my case, I've had people both from minoritized groups and majority groups that really gave me opportunities and sat down with me at times when I was learning, I didn't know what I needed to do, and taught me what I needed to do, and spent Saturday mornings writing me recommendation letters so I could go to the places where I ended up going to, and graded my exams and taught me and sat down with me in office hours. And those are the people that I like to honor today, because those are the people that got me to where I am, not the other ones. That's number one. And number two, I found that I like to give people the opportunity to change. I don't think that our experiences are static. So I've had people that have doubted me and then supported me. So I think it's more nuanced that way too. And I think Nelson Mandela had a beautiful quote about that I don't remember at verbatim, but it was something along the lines of like, when you liberate yourself, you liberate the oppressor too, kind of thing. So I think I'm not saying that those experiences are not real. I'm just saying let's remember they're one narrative. They're not. When we pick a narrative, we're not picking another narrative. They're both true. So let's not forget the other one, because the other one is the one that's going to take us where we need to go.
Thank you. I'd like to ask one more question, and this is from STEM Starter Academy. Can you tell us, why did you start Ciencias Puerto Rico, and how can we connect with that organization? Is there a way we could maybe start an exchange or begin to exchange between 4Cs and your students that you're working with there?
Yeah, well, absolutely. So I started Ciencia Puerto Rico when after I have finished my PhD. So, and I was in another part of my training, which is called a postdoc, which is right before you become faculty. At that time, I was in California, in Stanford, and I had been in science long enough that I had met a number of Puerto Ricans. Well, there was no organization that kind of brought them together. I didn't know where they were, and but every time I would go to a conference, I would meet one or two, and then we'll talk, and we'll be like, "Oh, it would be great to stay connected," but then we just would go our separate ways and not stay connected. So at that time, I thought, we have the internet, and this is before Facebook actually started since Puerto Rico. We have the internet, and I was thinking maybe that's an easy way of bringing people together. And there was another grad that had created a database for reagents in the lab that I was in, and I convinced him to help me change the database so that people could input information about themselves rather than the reagents. And I launched it, and I thought at the time maybe, I don't know, 25 to 50 people will sign up. And I define it as people interested in science in Puerto Rico, and right now it has close to 14,000 members. So it's very big. And it's a bilingual depository of information about science. So people should go and check it out. If you write Ciencia, which is CIENCIA, Ciencia is science in Spanish. If you forget, just go to Google Translate and write science and translate it into Spanish. Ciencia and then Puerto Rico, you find it. You can write those two things in Google and you find it. It's free, and it's essentially a resource. It has a bunch of profiles. It has information for educators, for scientists, for students, in a bilingual format. And what was inspiring about it was to see the community that kind of like coalesced around it, and how people started collaborating online to create projects that impacted education and science.
Thank you. I will look into that on behalf of STEM Starter Academy and connect with you there. We've got another question in the chat. I read an article recently about spiders offloading some of their memories into their webs. Did you read this? Any thoughts?
I haven't read it, but I have a friend in Johns Hopkins University that's studying how spiders make their webs, which is just really, it's really fascinating. I wouldn't say offloading their memories, I think it's just, I didn't read the article, but I would imagine it's just more metaphorically, like it's not literally they're putting all their memories into the web. It's more like somehow they have that representation of how that should look like in their brains, and then they build the web based on that. The same way that you can think about your house, and it's an architecture that lives in your brain. If I were to ask you right now, if you exit your room and you look to the right, what do you see? And most people here will be able to answer no problem. So similarly, and so it lives in your brain, but it's not a real space in your brain. So similarly, imagine now you're an architect and you build that. So you're offloading that concept into a real space. So that's what the spider does. And there's another question that we don't understand anything about. Like how is it that the spider knows where it is in space? How does it measure? How does it decide? Because it doesn't, you haven't seen a spider web that just kind of floats in the air when it was built. I mean, if people break it, yes, but when it was built, so the spider knows exactly what type of web is going to fit there and where to build that and everything. It's just fascinating. And how, who taught the spider that? He didn't go to school or community college or university to do that. How did it learn? It's fascinating.
Thank you so much, Dr. Colón-Ramos. I'm going to turn it over to Asher to wrap it up today. Thank you so much.
Thank you so much, Dr. Colón-Ramos. I have learned so much this afternoon spending this moment with you. I could probably just sit here and listen to you for hours, because I think it's like an illumination. It just keeps opening up more and more questions. I would like to say thank you for bringing your authentic self to us here at 4Cs. Not often does that happen, but when it does happen, we recognize and we say thank you. Thank you for showing the growth and development of your beautiful 3 girls. Thank you so much. I also want to say thank you for sharing your research with us. I almost felt like I was brought inside of your lab with your stories. I want to say thank you to Bridget Berger, director of the STEM program here at 4Cs. The 4C, the 4C Starter Academy, the Cape Cod Regional STEM Network, and the NSFS Scholar Program. As a point of concern, registration for our 4Cs returning students is November the 2nd. It's right around the corner. Please get a hold of your advisors so that you are taking the courses that you need to take and that you're sailing right into spring 2021. This is extremely important. The November 3rd election next Tuesday. Please vote and vote. Our last program of the year is coming up, and it's November the 2nd. It's called Playing with Fire. We have two exciting poets that are going to come to our campus. It's called From Coast to Coast, Poetry Matters. Register to enjoy a beautiful evening of poetry and storytelling. We have Annabelle Garcia Torres and Peggy Robles Alvarado. Oh my goodness. Woo! It's fire. It's going to be in that room on the 2nd. Please join us. The link will be sent out this afternoon, and I hope that you sign up and get your seat. I would like to say thank you to our technical staff for producing a wonderful program like what we've had here this afternoon. This is a collaborative joint effort between the Engagement Office and our STEM Starter Academy. Thank you and have an absolutely wonderful afternoon.